Our series. I'm Glenn Garrett, director of the EvoS program, and we have a really special guest in Andrew Gallup today. Before I formally introduce him, I have some housekeeping details I'd like to apprise you of. If you are a student in the psychology department subject pool, to get credit, we have our good friend Alana up in the corner. She's waving right there. She has forms that you can fill out, so please make sure to catch her sort of at the end, and if you haven't already, and we got the form for that. We've got a couple other announcement kinds of things. The summer online courses are up, available, and open and ready for business. There are several EvoS courses. The entire EvoS program is available online in the summers. So if you do want to take the best program on campus online in the summers, you are able to do that. And so we have lots of courses, including the first ever offering of Evolution in Human Health, which will be taught by Dr. Hamilton Staple, who spoke last week. And that's going to be a very unique opportunity for our students, so we hope students will take advantage of that. Couple other events coming up. On Thursday, there is going to be a discussion. I'm saying discussion, I'm using the quote thing, that I'm going to have publicly with Andy McIntosh, who is a physicist from England, and I was asked by Adam Gordon, who's an alumnus of ours, who also organizes Christian-related events, and so it's kind of going to be a discussion related to evolution and creationism. It's going to be 8:30. I will be summarizing a lot of psychology research that I think helps speak to the utility of evolution, and my mom thinks it's a good idea for me to do this. So come on out on Thursday at 8:30. It should be fun. The final speaker in our seminar series is going to be Dan Glass. He will speak next week in this room at the same time. Dan Glass is an alumnus of our master's program. He has published, I think he has 8 publications or around that from his 2 years as a master's student here. His master's thesis on the evolutionary origins of obsessive compulsive disorder is absolutely top-notch research, and he will be coming to discuss that next week. I hope that you can attend. Additionally, Dan will give a liberal arts and sciences alumnus speaker series talk in this room at 3:15 next week. He's a very good example of a very successful New Paltz student who will be talking about what he did to succeed when he was here, things that you might want to take advantage of. And so he'll be essentially telling the story of his academic journey. He recently has been admitted to a clinical Ph.D. Psychology program at Suffolk University in Boston. So Dan's just a great guy. He's a great researcher, a great presenter, so I hope you can come out for that. It is my pleasure to introduce Andrew Gallup. I first met Andrew when he was just a young undergrad himself at the first meeting of the NEEPS conference in 2007, and he's always impressed me. He's an extremely hardworking, sharp, and successful guy. He, after finishing his bachelor degree at the University at Albany, he got a Ph.D. in biology at Binghamton University, working closely with David Sloan Wilson on the Binghamton Neighborhood Project. In putting my introduction together, I said, I remember you had a lot of publications in grad school. How much is it, like 10 or even 15? He said, you don't have to mention it, but it was about 20. So Andrew is an extraordinarily successful guy. He's very efficient in his work. He's published on lots of different areas. He is associate editor for the Journal of Social, Evolutionary, and Cultural Psychology. After finishing his PhD, he did a postdoctoral research fellowship at Princeton University in the Collective Animal Behavior Lab of the Ecology and Evolutionary Biology Department. From there, he has gotten his current position as visiting assistant professor at Bard College in the psychology department. So I think that Andy's really a very good example of someone who crosses disciplinary boundaries with his interest in evolution as a common and core theme. And I found out today, I was very happy to find that he has just accepted an offer in the psychology department as a tenure-track professor at Oneonta, and he will be talking about the evolutionary underpinnings of yawning. So please join me in welcoming Dr. Gallup.
Okay, can everyone hear me in the back? Great. I want to thank you especially give my thanks to coming out to this talk with the wonderful weather outside. It's difficult for me to be inside currently, so I appreciate you coming in to listen to this talk today. It's just an honor to be here as part of the New Paltz EvoS seminar series. So the title of my talk, Why Do We Yawn? Integrating Neuroscience, Physiology, and Evolutionary Theory. First off, we need to start off with a definition of yawning. It can be defined as a large gaping of the mouth, accompanied by a longer inhalation and followed by a shorter expiration of air. Okay? Before I go any further, I want to provide a warning to you all that you may yawn during this talk, and you may have already yawned. You may continue to yawn. And for that I apologize, particularly if it's discomforting. But I will not feel upset by any means if you continue to yawn. It will not be a sign of disrespect to me, and I hope you understand why before the end of this talk. But just in case you haven't yawned yet, maybe you could start things off. That was a good one. Okay, so why do we yawn? I became very interested in this topic as an undergraduate when I realized, after a pretty short investigation of the literature, that there's no consensus on why we yawn. Everyone yawns on a daily basis and multiple times, frequently in the early hours of the morning and the late hours of the evening. In many cases, it goes overlooked. Okay, so why do we yawn? Hopefully, recent research has begun to shed a little bit of light on that. To take an evolutionary perspective on yawning, I need to mention the pioneering work of Nicholas Tinbergen and his 4 considerations. And these will be considerations that I touch on throughout my talk. Okay, the first being the phylogeny of a particular behavior or trait, and I will discuss that, the phylogeny of yawning behavior. Then the ontogeny, how it develops, how it works, what are the approximate mechanisms of this behavior, and then from an evolutionary perspective, what tends to be focused on is the ultimate explanation. Okay, what function does it serve? So my talk's going to be set up into 2 primary sections. First, I'll describe these 4 major considerations for spontaneous yawning, yawning that we do in isolation, the yawning that we do when we're not cued to yawn by others, and then following that, we'll go through these 4 considerations for contagious yawning. Okay? As you'll notice, there are different phylogenies, ontogenies, approximate, and ultimate mechanisms for each. But to begin with spontaneous yawning, has been documented in all classes of vertebrates. Okay, there's at least reports documenting the incidence of yawning in fish, amphibians, reptiles, birds, and mammals. However, a more thorough investigation suggests that it is only mammals and birds that yawn the way that we categorize and define yawning, although similar action patterns have been observed in these other species. Okay? So, if you didn't already know, other mammals and even birds yawn. And I'll show some video footage to demonstrate that fact. Next, the ontogeny. When does yawning develop? Well, in humans, it actually develops before birth, okay, in utero, and as early as 11 to 20 weeks gestation. So I'm going to play an image here of a fetus yawning. You will want to focus on this area. I'll play it a couple times because it takes at least one viewing to orient yourself to actually witness the yawn. See this gaping mouth right here? Okay, so this is the face. Plays again. Gaping mouth right there. Okay, so we yawn very, very early on in life, suggesting that it is a very evolutionarily conserved behavior. okay, occurring so early on in our uterine development suggests that it is in fact phylogenetically old, okay? If we think about the proximate mechanisms of yawning, the neuropharmacology, research has demonstrated that it is under the control of a number of neurotransmitters, in particular dopamine, acetylcholine, serotonin, oxytocin. These all facilitate yawning or increase the frequency of yawning. However, there are also some neuropeptides, in particular opioids, that have been demonstrated to inhibit yawning. Okay, and while we do have a much larger understanding about the neuropharmacology of this behavior, this research is very recent, has only occurred in the last few decades, and there's still much more to be known. As far as identifying where yawning is under control as far as the anatomy of our brain, it has been documented that yawning is under the control of the PVN, okay, of the hypothalamus, the paraventricular nucleus of the hypothalamus. Okay, the hypothalamus is often called the body's thermostat. It maintains homeostasis by constantly assessing temperature, blood pressure, levels of oxygen, glucose in the blood, a number of other factors. Okay, the presence of the control center of yawning being within the hypothalamus suggests that it is indeed part of our involved in some sort of homeostasis, okay? And therefore, spontaneous yawning may have some homeostatic function. But before we get there, I want to talk a little bit more about some of the proximate understanding of this behavior. Yawning physiology. So yawning occurs during low states of vigilance measured by EEG, okay? So placing electrodes on various portions of the scalp, we notice that electrical activity of our brain slows prior to yawning, okay, indicative of low vigilance. In addition, we see through electrocardiogram studies measuring activity on the surface of the brain, not the scalp itself, but the surface, we see that shows increases in arousal, okay, following yawning. This depiction here between these two, a control and a yawning response through the electrocardiogram, showing an increase in arousal. And in addition, yawning provides an increase in blood flow to the skull. The large gaping of the jaw, accompanied by a yawn, increases the flow of blood to the brain and increases also the flow of venous return from the skull. Okay? So this is the basic understanding of this behavioral phenomenon thus far. And there are a number of theories out there for why we yawn. Too many to count on fingers and toes, in fact. However, the overwhelming belief is that yawning functions to increase oxygen levels in the blood. Okay, that we yawn because we do not have enough oxygen in our blood or brain. Okay? However, a number of studies have actually falsified this hypothesis, okay? Providing support that altered levels of oxygen or carbon dioxide do not influence yawning frequency. So having individuals breathe gas that has an increased level of oxygen or an increased level of CO2 leaves yawning unaffected, does not influence its frequency. In addition, physical exercise sufficient to double breathing rates or respiratory rate has no effect on yawning whatsoever. And then lastly, respiratory period actually increases following yawning when, if in fact it was to promote the increase of oxygen, we would expect that the respiratory period should actually be decreased. So respiratory period is the time that it takes in between breaths. And after we yawn, we actually take breaths more infrequently. Okay? So therefore, to this point, there's little to no support that yawning has anything to do with blood oxygen levels. Therefore, I'd like to introduce a theory that was developed as my honors thesis just a few years ago, known as the brain cooling hypothesis or the thermoregulatory theory. Okay? According to this theory, yawning functions to counteract intermittent increases in brain temperature. okay, and that the physiological consequences of yawn may provide a cooling component. And ultimately, if yawning provides a cooling component, that could promote mental efficiency and processing, okay, by maintaining thermal homeostasis, okay. Brain temperature. Brain temperature is controlled only by three variables: the rate of blood flow to the brain, the temperature of that blood flow to the brain, and then the rate of metabolic heat production that the brain produces. Okay? In addition to yawning, brain temperature is controlled by the hypothalamus. The physiological consequences of the yawn, that being the deep inhalation of ambient air temperature and the muscle contraction of the large gaping of the jaw, could function to promote cooling in the first two levels. Okay? Taking a deep inhalation of ambient air temperature in this room is about roughly 70, 72 degrees Fahrenheit. That's a pretty large difference between our running brain temperature, being close to 98 or 99 degrees Fahrenheit. Therefore, taking a deep inhalation of this air may provide some cooling by cooling some of the oral and nasal orifices in which the air passes through. And interestingly, there's a strong presence of venous return in these surfaces that could be altered by this exchange of air, that couples very closely with the arterial blood supply that's supplying blood to the brain, which could act to cool the arterial blood flow. In addition, this gaping of the jaw, as I've already mentioned, increases the rate of blood flow to the skull. So I've already kind of touched on this, but the processes of cooling would be the enhanced blood flow to the brain, countercurrent heat exchange with ambient air temperature, and then thirdly, in humans, as well as some other species, including avian species, ventilation of the sinus system. Okay? The contraction of the jaw muscles acts to flex the paranasal sinus, okay, which promotes the ventilation of this sinus system and may promote the evaporation of sinus mucosa. Okay? So there are a number of ways in which physiologically it is certainly very possible that yawning decreases brain temperature. Okay? However, this would need to be confirmed. There are three primary ways in which I've gone to investigate this brain cooling hypothesis. The first is by investigating whether or not behavioral methods of cooling, there are a number of which we can look at, alter the yawning frequency of the individual. Second, and most directly, we can assess whether or not there are any distinct brain or body temperature changes that surround yawning events. That would provide the most clear evidence. And then third, any thermoregulatory behavior should be influenced by the ambient air temperature around, okay, for homeotherms that maintain this internal constant body temperature. So to begin with the first mode of testing, the methods of behavioral cooling, it turns out that deep nasal inhalations, okay, and exhalations, so breathing exclusively through your nostrils, can reduce temperature in areas of the brain, and in particular, the frontal cortex. So deep nasal inhalations, in fact, can reduce temperature in the frontal cortex by as much as 0.8 degrees Celsius. That's about 1.5 degrees Fahrenheit. In addition, simply cooling the forehead can provide a measurable degree of cooling to the brain by cooling facial veins, which is then transferred to the dura mater. Okay? This has been shown to cool the brain by degrees of 0.2 to 0.3 degrees Celsius, okay, which is quite substantial, accounting for roughly 80 to 90% of the circadian variation in brain temperature. Okay, so while 0.2 and 0.3 degrees Celsius sounds very, very small to us, it's actually representative of very large change as far as our circadian change in brain temperature is concerned. So what we did for my honors thesis, in fact, we brought students or participants into the lab and we altered their breathing rates, breathing manipulations, or we altered their forehead temperatures. Okay? So the breathing conditions, we had participants breathe exclusively through their nose, exclusively through their mouths, exclusively through their mouths while wearing a nose plug, or we just brought them into the lab and didn't even mention breathing whatsoever, serving as a control. Most individuals will inhale through their nose and exhale through their mouth, or they may show some fluctuations in the way they breathe on any period of time. But we wanted to serve that as a control, and we were anticipating there would be a mixture of nasal and oral breathing in that condition. So the hypothesis was that nasal breathing, if it cools brain temperature, individuals should be less likely to yawn if they're breathing exclusively through their nostrils. So while performing these breathing actions, we had the participants watch a contagious yawning video. Okay, a video that was one of the segments I clipped and showed you earlier. Okay, it was a mixture of individuals yawning, individuals having a neutral display, and then individuals either smiling or laughing. Okay, intermixed. It's about 3 minutes long, and about 1 out of 2 people who watch this video under no experimental manipulations will yawn in response to viewing it. Okay, and what we found. The white bars represent the number of participants yawning, and the black bars represent the number of yawns that were observed. We found that in all other conditions except for the nasal breathing condition, between 40 to 45% of our participants yawned while viewing these videos. So there were 11 participants in each condition. Okay, but in the nasal breathing group, not a single participant yawned. For the forehead manipulation, we had between subjects design, so different participants would come into the lab and they'd be asked to either hold a cold pack, a warm pack, or a room temperature pack to their forehead. Okay, and then we measured yawning frequency thereafter. The cold temperature pack was soaked in ice water and was maintained at about 40 degrees Fahrenheit before they put it on. The warm temperature pack was 105 degrees Fahrenheit, I believe, and the room temperature pack was between 70 and 75 degrees. Okay? Under the same conditions, we see that about 40 to 50% of the participants yawned in the warm temperature pack and the room temperature pack, but only 1 out of 11 of the participants yawned in the cold temperature condition, and that person only yawned once. So therefore, only 1 out of 22 participants in the behavioral brain cooling groups yawned for just once, while in all other conditions, 25 out of 55 participants yawned, 45.5%, and they yawned for a total of 51 times. So there are very, very significant differences based on these two groups, these classifications. And the interpretation is that cerebral cooling stimulated by the nasal breathing and forehead cooling conditions inhibited the mechanisms that would normally trigger yawning. Okay? But that's just an indirect measure of brain temperature. Okay, holding a cold pack to your forehead or altering your breathing rate. So we sought to more explicitly test this hypothesis by measuring moment-to-moment temperature, brain temperature of Sprague Dawley rats, lab rats. Okay? Using thermocouple temperature probes, we implanted these probes in the prelimbic cortex of these rodents, and we simply put them in a testing chamber in which we video recorded them. Okay? And we were interested in observing whether or not yawns were accompanied by brain temperature fluctuations. And the prediction was that yawning should be preceded by rises in brain temperature. So yawn should be triggered by increases in brain temperature, and that following a yawn, we should see corresponding decreases in brain temperature if, in fact, the yawn does provide this cooling component. So what we found is just that. That vertical dotted line represents the exact point in which the yawn occurs. Okay, and this is averaged across a number of rats for an even larger number of yawns. And we see that leading up to the yawn, we see rapid increases in brain temperature, okay, on the magnitude of about 0.1 degrees Celsius. And that shortly after the yawn, after temperatures continue to rise and then begin to reside quite substantially. So we see corresponding decreases in brain temperature following these yawns. Okay? The overall cooling effect of the yawns on average was about 0.12 degrees Celsius, which represents about 25% of the circadian variation in brain temperature changes. Another important note is that for this study, all increases of 0.1 degrees Celsius or larger that we observed during our study corresponded with yawning and stretching time points alone. And all rapid decreases in temperature of 0.5 degrees Celsius or larger were followed yawns and yawns only. So the largest changes in temperature that we observed during our entire study were directly prior to yawns and stretches or directly after yawns and yawns only. There are a number of other ways to assess changes in temperature, body temperature, or localized body temperature. One way is through thermal imaging. Thermal imaging is becoming a really popular in the study of animal behavior. Okay, a way to measure physiology simply with a camera is just something that people would have never believed possible a few decades ago. Okay. So with some researchers in collaboration, we investigated whether or not there were changes in the surface temperature of the same species of rat before or after yawns. In particular, we were interested in changes in corneal temperature, okay, or the temperature of the eye, as that would be most indicative of internal temperatures, and in particular brain temperatures. And the numbers on this figure are very small, and I wish I could change them, but they're not in my hands. But basically, what we found is that in this image here is at the point of yawning, and thereafter we see decreases in corneal temperature on the order of 0.6 degrees Celsius. Okay? Suggesting that not only is brain temperature changing, but more localized, or generalized rather, changes are occurring in a relatively localized area, that being the skull, okay, following yawning events. So that the physiological consequences of this behavior do have profound implications to thermoregulation. I mentioned earlier that birds yawn, and in fact I've studied this popular bird here known as a budgie. Okay, or your common parakeet that you'll see in the pet store. These are the same species that I study. One way in which you can experimentally manipulate the brain and body temperature of a budgie is by physical restraint. So you can catch these birds and basically hold them against their will, and this stresses them out quite substantially. And we see that in just a few minutes, their body temperature measured under their wing, so placing a thermistor right under their wing and placing it against their body, we see their body temperature rise by 2 degrees Celsius in just 4 minutes of being held. Okay? So the prediction is, following this experimental manipulation, these birds should yawn more frequently, but in addition, if there's individual variance in how much they're stressed out, so not all of them increased to 2 degrees Celsius, but on average they did, we were interested in whether or not the individual variation in this change predicted their yawning behavior. And what we found is that birds that were more stressed out physiologically, who their body temperatures raised more substantially during this manipulation, those were the birds that yawned sooner following release. So the higher the brain temperature of the bird during the handling, the sooner they yawned following release into the aviary. Okay? Very consistent with the view that yawning may be a thermoregulatory mechanism involved in cooling. I'll return to this study a little bit later on. So this is fairly good evidence to suggest that yawning is involved in brain and body temperature changes in some other species. But what about humans? Unfortunately, taking direct measures of brain temperature is a pretty invasive methodology that we can't do on many humans in the lab. But shortly into this line of research, we began to be contacted by a number of individuals across the globe who had interesting yawning symptoms. And two of them had nearly identical symptoms, both reporting these frequent and debilitating bouts of excessive yawning that they experienced on a daily basis. So these women, aged 39 and 61, both reported of these uncontrolling yawning attacks that lasted anywhere from 5 to 45 minutes in length, where there was just repeated yawning. They could not stop yawning. And that these attacks could occur as frequently as 1 to 15 times per day. They were completely debilitating. Neither woman could work. They were both very conscious of even going out into public because they were afraid that they would have these yawning attacks. And not only were they physically debilitating, but they were embarrassing to them. And they had seen a number of physicians and specialists to try to diagnose these symptoms. And they'd both been ruled out from having sleep disorders, and along with a number of other things, epilepsy, multiple sclerosis, and neither of them no doctor could identify what was causing their yawning. So we asked them to take a survey to assess some of their medical history as well as to investigate whether or not their yawning was accompanied by any identifiable body or brain temperature changes. One of the women could identify or predict the onset of these symptoms. Okay? So there was some change that she could detect that allowed her to anticipate that the attack was coming, and then she could act appropriately. Unfortunately, the other woman did not have this ability. But for the one that did, we asked her to carry around an oral thermometer with her. And when she could anticipate the attack coming on, to take her temperature. And then when the attack was over, to take her temperature again. And what we found is that leading up to the yawning attack, that this woman experienced mild hyperthermia. Okay? Her average oral temperature was 37.5 degrees Celsius. 98.6 would be 37.0. And that following these yawning attacks, these excessive yawning bouts, that her oral temperature decreased on average by 0.4 degrees Celsius. Okay? While again, this is not a direct measure of brain temperature, it is representative of localized changes in the skull, oral temperature. Okay? So, suggestive that this is not simply a product or simply a thermoregulatory component in animals, but also humans. I've also recently begun collaborations with an individual in Europe who's using the same thermal imaging technology I mentioned for the rats to investigate whether or not similar changes are accompanied by yawning in humans. That research is underway. Okay, so that represents the brain and body temperature changes that are accompanied by yawning that we know about thus far. The third and final way to test this is through changes in ambient temperature. Okay? So according to the brain cooling hypothesis, there should be a thermal neutral zone in which ambient or yawning frequency is relatively infrequent. Okay? What we'd experience now, okay, where generally our thermoregulatory mechanisms are not coming into play. We're very good at maintaining 98.6 degrees in a room like this. Very good. Okay, we don't need to sweat. Our thermoregulatory behaviors don't really need to kick in. And according to this hypothesis, we'd suggest that as ambient temperatures begin to drop, that yawning frequency should go down, as cooling mechanisms should be less frequent in general, and very low temperatures may provide some sort of thermal shock, okay, may be maladaptive. But going in the other direction, as ambient temperatures rise, we'd suspect that we should see increases in yawning frequency as well. But perhaps counterintuitive to this hypothesis, I would suggest that as ambient temperatures continue to rise and approach body temperature, that we should see decreases in yawning frequency. And the reason is that as ambient temperatures approach or exceed body temperature, taking a deep inhalation of that ambient air would not result in cooling. And if ambient temperature was indeed above our body temperature would result in the opposite of cooling and promote heating. Okay? So these are very basic predictions that can be investigated. I first began to study this in parakeets. They served as a really great model. Parakeets are a wonderful test animal for studying yawning because if you just line up the perch to be perpendicular with the camera and you have a plexiglass window in front of them, they will just stare right at the camera for hours. It's very easy to identify when they yawn. So what we did is we built this thermal chamber with this plexiglass window where the birds were watching our camera and we were watching them. And we changed the ambient environment in a number of ways. We first kept it constant to serve as a control, and then we increased the temperature very rapidly from 72 degrees Fahrenheit to about 90 to 92 degrees Fahrenheit in just a period of 20 minutes. So we increased the temperature on a degree Fahrenheit per minute. And then in addition, we held the ambient temperature very high, just below their body temperature, between 94 and 98 degrees Fahrenheit, to see how that would influence their yawning. And consistent with the thermal window hypothesis, we found that we see the highest rate of yawning during this rapid increase in temperature. But as ambient temperatures are held close to body temperature, we see a drop in yawning frequency. Now, while yawning frequency drops at this time, other thermoregulatory mechanisms, more well-documented thermoregulatory mechanisms, kick into play quite substantially. Okay? So these birds are displaying evaporative cooling, gular fluttering or panting, and they're also venting their wings to promote evaporative cooling. Okay? But their yawning frequency drops. But from this study and from this study alone, it would be difficult to determine whether it was rises in temperature that triggered yawning or any change in temperature, right? So we conducted a follow-up study which we replicated the same increases in temperature over a 20-minute period, and then we had another condition that had corresponding decreases, the same range of temperature but decreasing it over a 20-minute period. So we built in a ventilation system to our thermal chamber, so after we heated it up, we could quickly ventilate it. Okay? And what we found is that we see the highest rates of yawning during the intermediately high ranges of ambient temperature increases. But during the corresponding ranges of decreasing temperatures, yawning rates are relatively unchanged. Okay? So it is not in fact changing temperatures, but it is increasing ambient temperatures that trigger this effect. Similarly, we can look at stretching as a control behavior, right? While yawning is correlated with ambient temperature and the other two primary thermoregulatory behaviors in this species, stretching is not correlated with these variables. So it is not that there's an increase in activity or increase in other types of behaviors associated with rising temperatures, but in fact, only thermoregulatory behaviors. So that was in birds. What about humans? I did a study in Tucson, Arizona, in which we were measuring the yawning frequency of pedestrians during different seasons of the year. Tucson, Arizona is absolutely beautiful. If you ever visit, or if you're ever thinking about visiting, you should definitely go. It is literally surrounded on all sides by these beautiful mountain ranges. Okay? Maybe not go in the middle of the summertime, however, because it can be quite oppressive, but it is a beautiful area. So what we did is we approached these pedestrians in the early summer months of June and then again in the winter months of February. And we asked them to participate in a survey that was trying to elicit contagious yawning. So they had them view images of people yawning. Okay? And in this study, we assessed not only the ambient temperature of the two different seasons, but also the relative humidity and some of the behavioral variables of the participants, their amount of time spent outside prior to testing, as well as the amount of sleep that they received the night before. And that we see that the sleep they received the night before did not vary between the two conditions. Ambient temperature, humidity, and the amount of time they spent outside did. Ambient temperature was higher in the summer, relative humidity was lower in the summer, and the amount of time they spent outside prior to testing was lower in the summer because it was very, very warm. Okay? And in fact, the ambient temperatures between the two conditions in the winter is 22 degrees Celsius. That's about what we're experiencing right now. So February in Tucson is pretty nice, while June represents temperatures at 98 degrees, 98.6 in fact, on average during our testing period. Okay, so pretty warm. So the prediction here was that we should see relatively high rate of yawning frequency in the winter, but we should expect a lower frequency of yawning in the summer, as taking a deep inhalation of ambient air that warm would be counterproductive as far as cooling is concerned. And that's exactly what we found. The rate of yawning dropped in half, okay, in the warm and dry months of the summer. Now, multiple regression analyses were employed, and in fact, it was not the humidity that was contributing to this effect. Temperature and time spent outside were the only significant variables. So that higher temperatures, we saw less yawning, and the more time spent outside in the summer, prior to testing, the less yawning we found. So recently, we've decided to try and test the other side of the thermal window hypothesis, whether extremely cold temperatures or winter temperatures in a real winter climate would reduce yawning frequency. And our study site this time was Vienna in Austria. And thus far, this study is still underway, but we've collected a decent amount of winter data. The winter's behind us now. And the average temperature of our data collection was about 0.4 degrees Celsius, okay, which is about 33 degrees Fahrenheit. Okay? So far, only about 2 out of 10, or 1 out of 5, just slightly over that, yawned in response to seeing our yawning stimulus. If we compare that to some of the rates we found in Arizona, we see that the cold temperature range is very similar to the very high temperature range, and again, very lower than that room temperature middle range. We're in the process of collecting more proper controls for the Vienna location, but I just wanted to present the figure with the Arizona data next to it. For the sake of time, I want to move to contagious yawning. So let's change gears. Going back to the considerations of Tinbergen, if we look at the phylogeny of this behavior, we come to very similar conclusions. Contagious yawning has been documented in some mammalian species and at least one bird species. Okay, chimpanzees, bonobos, gelada baboons. Some evidence, although it's controversial, in dogs, and then most recently in parakeets. Okay, yawn in response to seeing their conspecifics yawn. So if you've never seen a parakeet yawn, you're in for a treat. So I want you to obviously focus on the 3 individuals in the red brackets here. And I apologize that the quality of the video is not as high as it could be. But I'll play the video a couple times. Focus on the top 2 individuals and then the bottom yellow bird. Okay, I'll play that one more time. So the top 2 individuals go back and forth. And these are not chirps. We're listening to the audio. These are yawns. And then the one underneath comes in. So the evidence we have to suggest that it's socially contagious is through a temporal analysis that we've done on these naturalistic flocks, although experimental studies are in the works. As far as the development of this behavior in humans, while spontaneous yawning occurs in utero, contagious yawning doesn't really begin to occur until the individual is about 4 years of age. And then it really begins to increase, and children become much more susceptible to catching yawns. There's another really critical psychological aspect that occurs right around the age 4, and that is theory of mind. Okay? If we look at some of the proximate mechanisms of yawning, some researchers have argued that it may be due to mirror neuron activity. So mirror neurons are neurons that fire both when the individual acts and when the individual observes the same action in another. So by grabbing this banana, there would be a certain neuronal activity in that primate's brain, and the same activity would fire when that primate watched that banana be grabbed by another. The same would be true in different scenarios, And it's been documented, these mirror neurons, in fact, have been documented in the 1990s, the last couple decades. Initially, it was in primates that they were observed, and since then we've been able to develop pretty good support that there may be a human mirror neuron system as well. Okay, so brain imaging studies using fMRI data have been able to document a mirror neuron system in humans. And in fact, these same regions of the brain are activated when individuals in fMRIs are viewing yawns. So viewing yawns does activate this human mirror neuron system. In addition, contagious yawning has been strongly linked with empathy. Okay? Higher levels of empathy, individuals with higher levels of empathy are more likely to yawn in response to seeing others yawn. And studies have shown that when viewing yawns or listening to the auditory sounds of yawns, the areas of the brain involved in empathy light up. Okay, so in particular, the ventromedial prefrontal cortex lights up, and in addition, other areas of the prefrontal cortex, the cingulate and prenunculus light up as well, areas that have been implicated in empathy. More behavioral research shows a very compelling association between yawning, contagious yawning, and empathy. So the degree to which we are or the susceptibility we show to yawn in response to seeing others yawn is closely linked to the social bond that we have with them, with the target yawner. So we're much more likely to yawn when kin and close friends yawn in comparison to simply acquaintances or strangers. Okay, suggesting that a degree of familiarity is associated with this response. Similar comparative research has shown that in chimpanzees, they're more likely to yawn when showed videos of their own in-group members yawning in comparison to out-group or stranger chimpanzees yawning. Again, showing this link between a group affiliation. So I was curious, how far does this group affiliation link go? In particular, are we more likely to yawn in response to seeing similar-minded political figures yawn in comparison to out-group political figures? So some fMRI research has shown that certain areas of our brain are activated when we see like-minded or congruent political figures, just their images, no yawning. Okay? But in particular, Democrats showed significantly more signal in their medial prefrontal cortex when viewing images of John Kerry's face in comparison to Ralph Nader's. Okay? This area is associated with positive emotion and empathy. Okay? So individuals neurologically appear to have more positive emotion and empathy towards like-minded political figures. Could that be true of contagious yawning? Currently performing a study right now in which I'm investigating this question, using primary targets of Bill Clinton and George W. Bush. And what I have is the participants brought into the lab, and they're viewing images of these figures, and they're listening to either breathing sounds or yawning sounds. So some yawning sounds would be like this. I don't know if that's playing. I guess I don't have the audio. Hang on one second. I don't know. It's very, very subtle. Sorry about that. There we go. Thanks. Just like that. So they'd be listening to with headphones, so it wasn't as subtle. It was actually very overt. They'd be listening to headphones, and this would be what they were listening to while viewing these political figures. And at Bard College, if you don't know anything about Bard College, it happens to be a very liberal environment. So it's not necessarily a representative sample, but I have some preliminary evidence to show you. So of the 30 or so, or just under 30 participants we have so far, most of them being liberal, the liberals are more likely to yawn in response to hearing yawn sounds while viewing Bill Clinton in comparison to hearing yawn sounds while watching George W. Bush. We have 5 identified independents. We would expect that the independent should be equally likely to yawn in response to either one. So that is very consistent. And we just have one self-identified conservative who yawned once to both. So we'd like to replicate or get larger samples elsewhere with a larger conservative population. But as far as some of the ultimate mechanisms of contagious yawning, I want to mention one other hypothesis, and that is that contagious yawning may have evolved to promote group vigilance. So based on what I've talked about thus far as spontaneous yawning and the physiological consequences of it, if spontaneous yawning cools the brain, and brain cooling is associated with increases in mental efficiency and cognitive capacity, which it is, then potentially the spreading of this behavior within a group could enhance the vigilance of that group. One way to study mental processing is through a number of cognitive tasks. This one happens to be what's known as the AXCPT task. Basically, it's just targeting your working memory performance. Okay? We have people perform this test, and intermittently we show them images, or rather not images, but videos of yawns. And then we assess their performance leading up to yawns and then following yawns. And in particular, we found that the reaction time on this task was significantly lower following yawns, when they yawn themselves. Lower reaction time would be better, okay, that improve cognitive performance. In addition to reaction time, we can measure accuracy. Following yawns, not only are they faster in their responses, but they're more accurate following their responses, providing evidence that yawning is indeed correlated with increases in cognitive efficiency, mental processing efficiency, consistent with the group vigilance hypothesis. Going back to the study of handling restraint in the birds, now in addition to just simply measuring the latency to yawn and body temperature, we're also measuring overall changes in yawning frequency over the course of an hour's time following this encounter, which is very similar to a predator encounter, okay, in these birds. And what we notice is a really interesting temporal change in their yawning frequency in comparison to a control. They show a significantly lower rate of yawning the first 20 minutes after being captured. Although the ones with higher body temperature yawn sooner, generally they yawn less the first 20 minutes after. And then we see this peak in yawning frequency 20 to 40 minutes thereafter before it returns to a general baseline level. Okay? So that provides a really interesting aspect. So I don't anticipate that if yawning is involved in group vigilance, that it is the best thing for you to do if you're presented with an immediate threat. Yawning would probably be very maladaptive in the face of an immediate threatening event. However, after a threat, so in nature, the incidence of a predator encounter are predictive of the next, either reduced likelihood that it will come or an increased likelihood that a future attack will come. In addition, contagious yawning may be beneficial in vigilance in the anticipation of future events. Okay? But this is measuring spontaneous yawning. So we performed another study in which we played these loud auditory disturbances, these bouts of white noise to these birds that really startled them. Okay? And then we measured the degree to which their yawning frequency, while in small groups, was temporarily clustered. So are there short bouts of high yawning frequency after these events, suggestive that they were socially contagious? The bars below the horizontal line represent significantly more clustering of yawning in time. The dark bars representing the noise condition, and the lighter bars representing the control condition. And in fact, we show that following this loud noise, yawning becomes more socially contagious in these birds. So again, trying to link this back to humans, does yawning become more contagious when primed to a threat to a group? So I brought participants into the lab and asked them to read vignettes of varying scenarios of depicting various threats. One of which, they were reading a story in which they were hiking down a trail and that they heard word through a walkie-talkie that they were about to encounter a grizzly bear somewhere down the line. Okay? I also had a scenario in which they were a number of different scenarios that primed them for threats. And I was interested in how this might influence their yawning immediately thereafter. Okay? And I want to show you that data right now. So immediately after reading about these threatening situations, we actually see a decrease in the proportion of participants that would yawn. Very, very similar to the pattern that we found in budgies or parakeets. We want to collect some data that would measure the yawning frequency 15 and 20 minutes after reading these vignettes. And the hypothesis is that that yawning frequency should rebound above baseline levels. We also were able to show that the higher degree the individuals rated these were threatening scenarios, the less likely they were to yawn immediately thereafter. Okay? So we're just about out of time. I just want to mention that there's some more ongoing research, comparative research, looking at group vigilance in yawning in chimpanzees, and then some general contagious yawning studies in ravens and marmosets. And I want to thank you all for listening today, and I want to thank some of my collaborators as well. Thank you very much. And I'd be happy to take some questions. Yes? Are people less likely to yawn? So there is an association between higher incidence of yawning with sleep deprivation. So sleep-deprived individuals, whether they be humans or other animals, are more likely to yawn. They show an increase in yawning frequency. One of the common symptoms of sleep deprivation is hyperthermia, elevations in brain and core temperature. So one of the primary functions of sleep is thermoregulation. So when we sleep, our brain and body temperatures are at their lowest point. And when we are held without sleep, this alters our ability to thermoregulate in subsequent days. And our brain and body temperatures are actually at an elevated point. So there is indeed a connection between the quality or quantity of sleep you have and your yawning frequency. But the underlying association between those two, I would argue, is thermoregulation. Yes?
Did you share your hypothesis with the two women that had the pathological yawning?
Great.
Is there any results from that?
Great question. Indeed we did. Yes. So we shared all of the results of our battery test that we collected a number of survey variables in addition to that experimental manipulation or tracking of temperature. And we've told them about our hypothesis, and we told them a number of ways in which they can do these behavioral cooling methods, deep inhalations of ambient air, or taking cold showers, or applying a cold pack to their forehead, and they've been able to reduce the negative symptoms that they experience. So just simply inhaling deeply through your nose can delay or completely abolish these attacks in some of these women, and same thing for holding a cold pack to their forehead. So at least on a day-to-day basis, it has provided some application. Yes?
If I could just share. In the first talk, I yawned about 8 times. Since you've told us about the breathing through the nose, I've been doing nasal breathing. I've only yawned twice.
Smart guy. Great job. I'm glad. Yeah, so yawning is elicited by a number of events, just increasing the complexity of the behavior and making it more likely that people are ever going to come to consensus about its true function. But one consistent trigger to yawning is stress and anxiety. So one common before public speaking events or before public performances, you'll see individuals yawn excessively. Apollo Ono, an Olympic speed skater, yawns excessively before each race. In addition, it's not uncommon for Olympians to yawn leading up to competition, and it's related to stress and anxiety buildup that is also associated with rises in core and brain temperature. Yeah, I think it's directly tied to the sympathetic nervous system and the autonomic nervous system. So, in times in which we are relaxing or revving down our system, it would be argued that there'd be a reduction in this behavior following, as long as that corresponds with the trajectory of our circadian temperature. And then when we're revving up this system, we may, leading up to that rev up, see alterations in yawning frequency. Another suggestion would be that if the sympathetic nervous system is powering down, that we might not be able to thermoregulate quite a bit, okay? And that you may see yawning frequency increase as well. Direct studies investigating the complexity of effects with the sympathetic and autonomic nervous system have not been conducted. But I certainly anticipate there's going to be strong correlations with a number of other physiological changes.
Thank you.
You're welcome. Yes?
Going back to the whole thermoregulation thing, yawning is meant to cool down the brain and the body temperature. What about times when you're doing large amounts of exercise? Because people don't tend to yawn during those times, yet your body temperature is raised during those periods.
Yes. So that's an interesting point. It's a great question. So it actually depends on the type of exercise that you're performing. So cardiovascular exercise, we see hardly any yawning occur. When there's increase in breathing rates, for instance, we see that people hardly yawn, although they're breathing like crazy and they're sweating like crazy. And I would argue that the reason they're not yawning at that time is because it would disrupt their respiratory rates, for one, and in addition, I believe that yawning is not a primary cooling mechanism, and instead is a compensatory cooling mechanism. It's an initial attempt to alter sudden intermittent deviations, such as was witnessed in the brain temperature figure, but that following extended periods of hyperthermia, such was demonstrated in the ambient temperature manipulations with the birds, increases their panting and increases the rate in which they're wing venting, but it decreases their yawning frequency. So yawning, in a sense, is an initial compensatory thermoregulatory mechanism and not a primary one. So I'm arguing that, in fact, that the changes in temperature that yawning would produce, while significant, are minimal in comparison to other thermoregulatory responses. Yeah.
That if it's an initial response to a quick stimulus, it's a somewhat delayed response.
Yes. So the change in brain temperature, I can put that figure back since we'll talk about it. Okay. So temperatures continue to rise for another 20 to 45 seconds after the yawn occurs. And basically, leading up to that yawn, we're seeing increases in brain temperature on the order of 0.1 degrees Celsius per minute. Following the yawn, we see that only begins to transition and go down after this short delay. Arguably, any type of change, convective cooling, is going to take time to transfer. And in addition, if brain temperatures are continuing to rise, some sort of mechanisms are in play, or physiological changes are in play that are making this rise, and that it is only after a period of time that you could actually counter that and begin it to go down. And when I'm saying initial, I think a minute leading up to or a minute following still qualifies as a pretty quick response. Yes?
So a fever is a time when the whole body has very elevated temperatures. Have you done any research on fevers and yawning?
It's an excellent question. Excellent question. So one question that arises is something very similar after I give a talk like this. So what about fever? Fever represents a raise in brain and body temperature Do we yawn excessively when we have fevers? No study has been done to investigate it. But I would make the other prediction. And I would argue, and I'd hypothesize, I have, that yawning should be decreased. The frequency of yawning should be reduced when we have a fever. So fever is different than elevations, irregular elevations in body temperature. An abnormal elevation in body temperature, an inability to thermoregulate, would be representative of hyperthermia. Fever, in a sense, while it does elevate our temperature, it doesn't represent hyperthermia, because a fever is actually representing an increase in our thermoregulatory set point. When we have a fever, it is because our hypothalamus increases the set point of our body and brain temperature to 100, 101, wherever it goes. And then our body and brain temperature reach that and then stay at that level unless it's increased or decreased further. But when we have a fever, we are thermoregulating at that newly revised set point. And it does not represent an instance of hyperthermia. And I've recently made the prediction that the use of antipyretics or fever-reducing drugs, I think then you should see, if you watch closely, very significant changes in yawning frequency. So if you have a high fever and you take some Tylenol or Advil or any other antipyretic to reduce that fever, I would argue that you should see an increase in yawning frequency shortly thereafter. So this antipyretic reduces the set point back down to normal levels, 98.6. But in the period in which the set point is set to that 98.6 and the time it takes to your body to actually go to that 98.6, you're going to see the trigger of thermoregulatory mechanisms, cooling mechanisms, to bring it down. And then I would argue, following the application of antipyretics, you should see a increase in yawning frequency until that antipyretic kicks in. But it's a very, very interesting question. And it remains an empirical question. Okay, so what are chi-square tests, and how are they used to demonstrate differences between groups? So chi-square basically measures, it's a non-parametric statistic that measures the proportion of participants that yawn. So you're basically comparing the proportions. In the studies of the seasonal climate variation, there was about 45% or 0.45, 45%, or about 1 in 2 participants, just under, would yawn in the summer condition, and about 1 out of 4 would yawn in the winter condition, and 1 in 4 would yawn in the summer condition. So we use a chi-square to see whether or not those proportional differences are statistically significant. With yawning, we often have to use these non-parametric statistics because we're dealing with simply counts, and it's a very infrequent behavior. So it's usually non-normally distributed. Yes? It's a good question. So the contraction or the muscle contraction in your face often squeezes the tear ducts. And that produces this tearing. Whether or not the tearing has any functional basis, or if it's simply a byproduct of the facial contraction, I don't know. But we can imagine that, although it need to be tested, that this tearing could promote evaporative cooling of the cornea. But that would need to be formally investigated. But it's a good question. It's a good question. It could be a byproduct of simply the motor action pattern, or it could have a functional basis.
Why don't we take one more question before the reception in CSB? You want it?
Yes. I would like to investigate it simply because of the possible association that yawning could have in cooling the paranasal sinuses. So sneezing would disrupt or alter the level of sinus mucosa, which may allow for more effective evaporative cooling. So therefore, we may suspect that following sneezing, we'd see an increase in a higher rate of yawning frequency than we otherwise would, in the absence of sneezing. That's a prediction that I would make, but no one has investigated it.
Thank you very much.
Thank you.